So we're going to try to keep this on time and actually leave 10 minutes for discussion at the end. Let me see. I'm John Alexander. I think I know most of you, or you know me. And we're going to talk today about actively reversible anticoagulation, a journey really at Duke, from bench and almost to the bedside. And our four speakers today are really going to cover this whole spectrum of research from the bench to the bedside. I'm going to introduce people. Bruce Sullenger, who's the head of the Duke Translational Research Institute and the discoverer of this technology, is going to talk. Then I'm going to give a brief overview of some of the early trials. Marianne Sellers, our project leader, is going to Marianne Sellers is going to talk about some operational lessons learned from our big Regulate PCI trial. And then Tom Povsic is going to talk about the Regulate PCI results. And we're going to have time for discussion at the end. So in case you can't tell who's speaking, these are everybody's pictures. I asked Marianne for a picture, and she refused. I tried to find a picture on the internet, and there are none. So she's been highly successful at grooming the internet. But this is the rest of us. All right, I'm going to turn things over now to Bruce. Bruce.
So what John asked me to do is sort of give you the background, the laboratory side of sort of this whole story, which again, Thomas was pointing out to me was 15 years of my life in a sense, so in 1 hour. So this is going to be kind of fun. So what I'm going to try to do quickly in 10 minutes is just give you sort of a background of how we started this project and how it led to all the clinical work we're going to hear about. So my group, which is actually in surgery here at Duke, was interested in making new therapeutics. Many years ago, David Savison recruited me here thinking about that sort of application. We got interested in these compounds called aptamers, which many of you probably know about now, but just quickly what they are a sort of a new or an alternative class of therapeutic where we're making RNA molecules that bind to proteins. And then the idea is they can inhibit the protein's function, similar in a sense to an antibody. And the way we make them is sort of outlined here, where we use combinatorial chemistry, like how well this pointer works, but doesn't work. But anyway, you just make a large library of different shaped molecules and you screen them for binding a target. And we got interested, again, 15 years ago now, and thinking about targeting different proteins involved in coagulation. We've made use this approach. You can actually make compounds against many different targets. We've made them against about 100. People have made them over 1,000 different targets now. But the main point of this slide is you can use that approach to get things that bind with high affinity to different proteins you want. Also, because you're doing that all in the test tube, you're doing this process in the test tube, you aren't limited to normal chemistry. So we generate things that are RNA molecules that are very stable to nucleases. So that lets you fairly quickly take them into inhibition assays in cell culture, animals, and then into humans, as you'll hear. And so, again, about 15 years ago, got interested in targeting things involved in thrombosis. And this is really because initially we were working on things to target antibodies involved in autoimmunity. And then people in surgery, particularly Bob Anderson, was encouraged to think about other indications which were much broader than insulin resistance, et cetera. And so we said, well, what are those? And everybody started to point out to us, well, thrombosis was something that the surgeons and the cardiologists were dealing with every day. And we were fortunate because Bob said, well, if you really want to go in this game, so we got interested in it, you need to go talk to the people over in DCRI. And so he got us in and he introduced us to Rob, Califf, and Bob Harrington here at that time. And we came over and we tried to get them excited and get their input about how to develop new therapeutics in sort of the space. And I remember, and I like to show this slide, as we showed a slide something like this, and we said, Rob and Bob, here's this new class of potential therapeutics called aptamers. They are like antibodies that what they really do is they block protein-protein interactions. And so this is a crystal structure, for example, one of these things we made to thrombin. You can see it doesn't bind the active site of thrombin, which is shown there in the red like a small molecule does. But what it does is it blocks thrombin from binding its substrates, its large molecular substrates. So I'll never forget at the time, I always joke with Rob and Bob, and I said, they kind of yawned. They said, why are you interested in doing this? Antibodies can do this. We're doing all these trials for small molecules in this space. You got to give us something more, make us excited about it. And I always say that we were either naive enough or glib enough at the time. We said, well, what's the problem with all the things that are being made that you guys are trialing. And they came back and it sort of surprised us at the time, and they said the issue is, and this is probably 2000, 2001, as you can see here, that the issue with a lot of these drugs was safety. And what they meant by that, I'll never forget the analogy that Bob Harrington really uses. He said, sometimes in the cath lab, I'll give one of these things, or one of our colleagues will give these things, and it's like sending out this email that you can't pull back. So you put this blood thinner on board, and then if you start to bleed, there's no way to reverse it. And so for a lab guy like me, that kind of made sense. And so they went on to convince us that, developing, and the people in this room know this, that developing drugs in this thrombotic anticoagulant space, you always run into this issue of major bleeding, and particularly they said that there was an unmet need to develop sort of controllable drugs, particularly in the acute care setting, so in the cath lab, the surgery suite, et cetera. So with this sort of charge in mind, we went back, they sent us away at this point. I always like to point out, I don't think they ever thought us to come back, because they say they told that to everybody. But we went back to the lab and we said, well, maybe this technology we're working on, this aptamer front, maybe a unique feature would be that we could make antidotes to it, to things that could rapidly reverse their activity. And if that was true, that compared to these other drugs that are being developed in this space, antibodies, peptides, or small molecules, you may have a potential unique feature and potential advantage. So went back in the lab, that was our hypothesis, and so we initially tested that with a couple aptamers, but the one that we moved forward most quickly is the one that we made to factor 9, the clotting factor 9. And so we're going to show you today sort of how we took that, made it initially, and moved it through the clinical studies. So we did that selection process I showed you, where we took 10 to the 15th molecules and we tried to find those bound to factor 9 with the highest affinity. This is the type of things you get. So these are all RNA sequences that you get out of that type of selection approach. I don't expect you guys to look at this or go through it, but basically what this tells you is something about the structure and sequence of aptamers you get out of the selection. And one of the things that you can see is, say, the AUA sequence was 100% conserved. So that was implied it was important, and you could look at this information and fairly quickly deduce what the minimum sort of link sequence was that was required for high affinity binding to your target. So that's this guy, which we called aptamer 9.3T. It's about 34 nucleotides. It actually is the precursor of the one that goes into the clinic. That one shortened, I think, another 2 nucleotides or so. You can see this guy binds with high affinity to factor 9, so 0.6 nanomolar. And at least for the preclinical studies, one thing that's nice about these type of things you can't do with antibodies or small molecules well is you can make negative controls. So we simply switched those 2 A's to G's to make a mutant version of the aptamer. And so that guy's dead now. And so it's very similar, but it doesn't bind anymore. So it's a nice control for your study. So this thing bound well. Now you can make this chemically, just like we do in oligonucleotides. So you can make kilograms of this thing, just like what you do with PCR. So it's different than an antibody. That's more like a small molecule in that sense. So we went on to ask simply in clinical clotting assays in normal human plasma, was this thing an anticoagulant, bound well, could it inhibit factor 9A's function? Just you can see here nicely, it does in a dose response. In an APTT assay, it has no effect in a PT, showing that you're specific to factor 9 function, and the mutant aptamer has no effect. Now, one of the things people always ask me when they look at this is you require higher concentration than the affinity. And that's because the aptamer also binds factor 9. So really what you're doing here is you're just titrating all the factor 9 in the plasma. So it's a 1-to-1 stoichiometry, and factor 9 is about 90 nanomolar, so you just have to add enough where you're inhibiting all the factor 9. So that was our first in vitro data in human plasma. Went on to ask, would this thing work as antithrombotic? This was work we did with Bill Fay a number of years ago now. Bill is at Michigan there, where he had set up this standard model of thrombosis where they expose the carotid artery of a mouse, they damage it with a chemical insult, and then they would monitor blood flow with a flow probe downstream, and they would put different things on board in the mouse and say, would it keep the vessel patent, basically. And what you can see is this is 0.5 mg per kg of this. Here we modified the aptamer, put a cholesterol on it. The reason why we did that is if you don't put anything on the aptamer, its half-life's about 10 to 20 minutes in circulation. Put a cholesterol on it, it ends up being about 2 hours. You'll hear later we put PEG on the thing, and then the half-life goes at about 24 hours. So depending on how you formulate it, you can change the clearance properties. And if you put nothing on it, it's renally cleared pretty quickly. And so in this case, he basically damages the carotid artery. You can see this is 0.5 mg per kg. he gets the mutant after all the guys that occlude in 10 to 15 minutes or so. And I think it was 9 of 11 animals he did. He saw no occlusion. And when you do histology, this is the type of data you would see. Here's looking at flow on the top with the wild type after. The blood flow remains pretty constant. There's no clot in the lumen. And then this contrasts the mutant guy. So this is our first data saying you could use this to block thrombosis. Again, this was published 10 years ago. But we want to get back to this idea. There are a lot of compounds that could do that. Harrington and Califf would point out to us, but could you stop that if you needed to? And so the first way we did this was fairly straightforward. We thought, well, if you had to adopt the structure to bind factor 9, maybe there was information encoded in that to design a second oligonucleotide using Watson-Crick base pairing rules. We all know from biology in high school that maybe you could make a second oligo shown here in red that would bind to that and take it from an active to an inactive structure. I don't have time to go through all the data. It turns out that works really well. We screened a bunch of them to this aptamer, and here's sort of a result from one of our first studies done in swine. I have to give, again, Bob Anderson a plug here, because we kind of got to this point and we said we have this interesting data in vitro, but we want to see if it works. Bob at that time in surgery gives $250K to go and make, this is before CTSAs, and you could get money like this quickly. He said, here's $250,000 from surgery's coffers, go make some of this and get some pigs and figure out we can do this. So we were very fortunate that at that time we had those type of resources lying around in surgery. But so in this study, what we did is, again, this is a cholesterol aptamer injected. You see in the filled squares, you can anticoagulant 0.5 mg per kg. You're quadrupling the clotting time. You're basically making the pigs hemophilic. And if you don't have an antidote, you can see this effect goes down over a couple hours as shown here. And then we took a second set of animals in this case, and we put a tenfold excess of the antidote at the 15-minute time point, and we looked 10 minutes later, and what you saw is it basically normalized the clotting time. So that was our first data from a large animal that showed that this idea of rapid onset but rapidly reversible anticoagulation could be achieved using this approach. And I have to say that this was the most surprising result of my career, because I thought it would never work this quickly when it was circulating really fast in vivo, et cetera. So we were encouraged, obviously, by that. The other thing I wanted to ask is, what about if you started to bleed? So this is a simple experiment we did in a mouse where you would dope either the mutant after or the wild type after, then you would challenge them by surgically clipping their tail and say how much blood you lost. And you can see, not surprisingly, you make the animals hemophilic, they bleed more in 20 or 30 minutes. If you gave the antidote immediately after cutting the tail, though, you would totally neutralize that effect. And that was, again, a little surprising to me because it meant it was very fast. So you weren't like getting some intermediate blood loss, you were basically reversing all of it. So that meant the on rate of the antidote was really quick in vivo. So at this point, we basically said, well, we want to try to see if we can do this in humans. This is animal stuff. So we started a company called Regatta. I should point out a lot of this work was done by Chris Rusconi when he was in the lab. And then Chris helped me found Regatta. He moved out there. We licensed the IP to Regatta, both for sort of these compounds, as I showed you, the factor 9 one, as well as a couple other ones, as well as this concept of antidote control. This led to these series of studies that we're going to hear about. So I'm going to hand it off to John.
Thanks, Bruce. Being able to work with Bruce and Chris Rusconi, who was then at Regatta, as we were thinking about these trials, was
I think I have one more slide, actually.
Very exciting.
This is just to point out we have a lot of other compounds, and if people want to think about developing against, and only a couple were licensed to Regatta.
Right. Yeah, really to any protein, you can make a small an aptamer inhibitor. So I'm going to now go over all the trials from the first human experiment up through RADAR in 10 minutes. So that's about a minute per trial. I'll just try to hit on the highlights of sort of the interesting parts of the design and lessons learned. So these are the trials. We did a phase 1A, 1B, 1C trial. We had less of an operational role in the phase 1C trial. We then did a little pilot, really led by Mauricio Cohen, who was a fellow here, is now at the University of Miami, and then the larger phase 2B trial, RADAR, and then Tom's going to talk about Regulate PCI. So in 2006, this was published, the first, and I think one of the interesting things about this story is the timeline and how long all this takes. So in 2006, the first trial our phase 1A experience with Reg1, now owned by Regatta. The trial was really led by one of our fellows, Chris Dyke, Rick Becker, Bob Harrington, myself, others were involved in it. And this was the first time we gave the drug to humans, and we did it in healthy volunteers. And actually the most interesting thing of the design and from a regulatory standpoint are the aptamer, the anticoagulant, and the antidote are here being developed as one drug. But they're obviously two compounds. And so you want to test the safety of each of them. So we actually tested the antidote first. Before we gave the aptamer alone, and then we tested the pair. And we wanted to do that so that if people had life-threatening bleeding, we obviously had some safety experience with that dose of the antidote before we gave them this aptamer. We studied a range of doses, and I think these remarkable pictures that Bruce alluded to in animals started to be replicated in humans. So you can see a range of doses, you raise the APTT, you give the antidote, and as fast as you can measure it, the anticoagulant effect goes away. This compound was now coated with a polyethylene glycol coat, which gave it its relatively long half-life. This is out 24 hours, about a 24-hour half-life of anticoagulant effect. We then replicated the same trial. This time we used fixed milligram per kilogram, but we used fixed dosing instead of milligram per kilogram dosing. And this time we did it in patients with established coronary artery disease who were also taking aspirin or aspirin plus clopidogrel. So we wanted to start to understand drug interactions, both pharmacokinetic and pharmacodynamic. Really, we studied a range of doses of aptamer and of antidote, and we used fixed double dosing, double the molar amount of the antidote to fully reverse the anticoagulant effects. And here again, we studied the anticoagulant, which was called RB006, and the antidote RB007. We studied it with antidote placebo and just all placebo. And again, you can see the same kind of pattern in patients with established coronary disease, not surprising, published 2 years later. And then also published that same year a study that we had less to do with, which I thought was really neat and fascinating, is we use variable doses of the antidote. So if you give, again here in healthy volunteers, people a fixed dose of aptamer and then give them various ratios of antidote, you can turn the anticoagulant effect all the way off, or halfway off, or 25% off, and you can sort of dial in the level of anticoagulation that you desire. All right, and then our first experience in actually patients undergoing procedures was this reversal PCI pilot led by Mauricio Cohen. Here we took a small number of patients undergoing elective angioplasty. We initially dosed patients with the anticoagulant plus glycoprotein 2B3A inhibitors, and then we partially reversed them, and then 4 hours later completely reversed them. Here we were concerned that acute reversal of anticoagulation might lead to thrombotic complications in somebody who had just had an angioplasty. That went well. We then did the same thing without the 2B3A inhibitor in 12 patients, randomized to Reg1 or heparin. And then we did the same thing, immediately reversing people 10 minutes after the procedure who had just gotten Reg1 on top of aspirin and clopidogrel. And again, this went well, and again, it was small numbers of patients, but we concluded that elective PCI was at least feasible with Reg1 with immediate reversal and immediate sheath removal. So this then led to the larger phase 2 trial RADAR, which Tom Povsic led, was published in 2011 in the European Heart Journal. And here we really had 3 objectives. And this was setting the stage for our phase 3 trial. The first objective was to verify that a 1 milligram per kilogram dose of pegnevacogen, that's now the name of RB006, which was the RNA aptamer, results in near complete factor IX inhibition. So we wanted to give a high dose that was really totally inhibiting factor IX. We thought we might be able to do this because we can turn it off when we no longer want anticoagulation. And then we wanted to determine the dose of enivamericin. This is the antidote, RB007, the RNA aptamer antidote, which when followed by immediate sheath removal results in a bleeding rate that's lower than unfractionated heparin, and then get a preliminary estimate of the efficacy of pegnivocogen in patients undergoing angioplasty. So regarding the first objective, these are data from baseline, immediately post pegnivocogen injection, right before cath, and post-cath in an ACS population. And this is the amount of factor IX inhibition that we achieve. So what you can see here is that with a 1 milligram per kilogram bolus of pegnivocogen, you essentially wipe out factor IX. You become a severe hemophiliac. So more drug is unlikely, what we learned from this is more drug's unlikely to cause a greater antithrombotic effect. In RADAR, we enrolled 800 post-ACS patients who had planned catheterization through a femoral access site. We randomized them to pegnivocogen or heparin, and this was an open label randomization. Blinding here is it was really complicated and we felt not necessary to the main objectives. In the heparin arm, the sheath was removed per standard of care after approximately 6 hours, and 200 patients were randomized to heparin. And then we randomized the pegnivocogen arm again in a blinded fashion to various amounts of reversal. So 25% reversal, 50% reversal, 75% reversal, or 100% reversal. And again, we were interested in how much reversal is necessary in order to be able to remove arterial sheaths. I had wanted to have a 0% reversal arm, but was talked out of that, which turned out was probably a good thing. All right, and we had bailout reversal for patients who had severe bleeding and planned interim DSMB assessments with plans to roll these patients out of the low dose reversal arm if they had bleeding, or the high dose reversal arm if there were thrombotic complications that were observed. Yeah, that's just those. So what we actually did was we ended up enrolling just 41 patients here because there was, on that first DSMB look, an excess of severe bleeding post-sheath removal with just 25% reversal. Those patients were then dumped into those other arms. And then these are the main results of the trial. So we used an acuity bleeding scale with acuity major bleeding shown in the gold bars and acuity total bleeding, which is really all total bleeding, shown in the blue bars. And here again, you can see in this low dose 25% reversal arm, there's more bleeding. This is an arm that was stopped early, only has 40 patients in it. And then in these other arms, you can see bleeding rates that are really similar to unfractionated heparin, which is over there on the far right. So we learned from this that you need at least 50% reversal, 75 or 100% reversal might get you a little bit better bleeding profile. And then intriguingly, again, this study wasn't designed or powered to look for an ischemic effect, but in the 473 patients who were randomized to Reg 1 compared to the 161 patients randomized to heparin, we saw a lower rate of death, MI, or urgent target lesion revascularization, essentially stent thrombosis, in the 30 days after their randomization. So an encouraging signal on ischemic events. Now, we stopped RADAR a little bit early. We actually, after about 400 or 500 patients were enrolled, this trial was enrolling in the United States, Canada, Germany, and Poland. There were a series of 3 patients that had anaphylactic-like allergic reactions. The first one was relatively mild. Patient got some hives and transient hypotension, recovered quickly. The second one was pretty severe, and we put enrollment on hold for briefly. And then the third one, which happened rapidly after soon after we had restarted enrollment, was also quite severe. And at that point, we had decided we learned what we needed to from the trial. We put enrollment on hold and did a number of other investigations into the etiology of these allergic events, which you will hear a lot more about from Tom. So we concluded that at least 50% anevomersine reversal was required for hemostasis in the early sheath removal, that pegnivocogen with partial or complete reversal resulted in numerically fewer ischemic events, not definitive by any means, and that these data supported doing an adequately controlled clinical trial. So with that, I'm going to hand the microphone over to Marianne Sellers, who was our project leader on Regulate PCI on this adequately powered randomized trial that we said we needed. And she's going to go over some of the operational lessons learned from the trial. Marianne.
Thanks to everybody for joining us today. I appreciate the opportunity to represent the operational group. Talk a little bit about some of the lessons that we learned from this trial. Even though you haven't heard the results of the trial, I think most folks know that we did stop early. So as are lots of our mega trials, this was a fairly complicated organizational structure that we had here. We had a lot of different groups that were involved. So the project management was done here at DCRI, but we also had project managers at C5 Research, which is at the Cleveland Clinic, as well as Parexel and Regatta. We did U.S. site management and shared that for the U.S. sites with the Cleveland Clinic. They also did our site contracts for us. We were involved with the Canadian Vigor Center, one of our academic partners, to manage the Canadian sites, and the rest of the world site management was done by Parexel. DCRI was responsible for data management, stats, safety. We got the first look of all the safety, and then the Parexel did reporting for the European authorities. We also were responsible for the clinical events committee, and that's sort of a long story, which we'll talk about. Want me to move? Okay. Okay. We also were involved in an angiographic core lab, which was at Mount Sinai Hospital. We had several vendors. ClinPhone did our IVRS. We had a commercial lab managing our allergic reaction samples, as well as all of our other biomarkers. We had 2 academic labs involved. The drug was done by Catalent, which is a subsidiary of Parexel. Parexel also did our study materials, and Stanford did our DSMB for us. And it was the first time that they'd ever done a DSMB, so that was a great opportunity with them. And Bob Harrington was our chair. So this is the list of folks who did the real work and have the real expertise operationally. So if you have any specific questions, you can check with them. So just to go through the different groups and the things that we learned, wanted to really say how important it was to have the faculty leadership that we did. Both Tom and John were so important in doing training for our internal and our external groups. As you saw, it's kind of a complicated and new science, so it was important for the teams to understand that really well so that they could help the sites understand what needed to be done. The faculty were always available. None of our questions were treated as if they were stupid, and they were really involved in developing the key processes for the operations, and that includes the CEC, which we'll talk about in a minute, the safety, the risk-based monitoring plans that we had. So they were so instrumental into making the project happen. Tom did our clinical helpline for us, and when I say Tom did it, I think 2 of the phone calls over the year of the project were taken by our fellow, who kind of appeared and disappeared. But Tom took every single one of those calls, and it was a little bit challenging because Parexel was in Europe, and they had what they called medical monitors, and they wanted their sites to call and talk to them first. And even though those people were cardiologists, some of the answers that they gave didn't quite always go with what we would have suggested. So I know Tom worked really hard to keep them up to speed. As far as stats goes, it was an open label study, which meant that the sites all knew what the patients were getting, but the folks at the DCRI were not allowed to know. The operational teams were not allowed to know what the patients were receiving, and that became quite a bit of a challenge, statistically as well as with the safety group, with the CEC. We had several communications with FDA, several calls. Those were really very helpful for the team to sort through how we needed to do things. We had these algorithmically adjudicated and manually adjudicated CEC events. And I think statistically that was really difficult for the statisticians to deal with as it was for the CEC. But we've learned a lot from it, so I think it'll be easier next time. We had a lot of biomarker samples, and that was really important as one of our endpoints, or several of our endpoints. And so one of the things that Jen, who was our statistician, wanted to know is how we set up protocols. Is it better to have these pre-specified windows where we have to have things and define those by the protocol, or should we have a little bit of a leeway because that's not always how things are collected in real life, especially in the cath lab? We also had, because Stanford was new to this DSMB stuff, we had a statistical liaison, which is a person who was a statistician who helped them to set up their processes and go through some of the data. And that was really a nice feature. The problem was FDA decided that that was probably not good because that person was a DCRI employee and that person was unblinded. So after we started that, we had to take that person away, although that was something that we would recommend for folks working with a new DSMB group. So CEC, as I mentioned, we had these algorithmically adjudicated events, which were our MIs and our bleeds. We had a whole team of all different groups that worked together to help define those and figure out how we can create those triggers. What ended up happening is we had about 85 pages of code to define those triggers for us, and it became so complicated we couldn't even tell if we were missing anything or not. So we really would recommend trying to make those triggers as simple as possible. We ended up reprogramming all those pages, and then we did get it much simpler, and I think that worked out better for us. We would also recommend that we share the definitions at the beginning of the trial when you're writing the protocol, because you write your triggers later and then you're sort of stuck with what you have written in your protocol. So you sort of need to think at the beginning. We had a lot of biomarkers and it made it really complicated to do these algorithmically and manually adjudicated events. One little minor thing was we had this CEC mailbox that had all these dashes, dots, hyphens in it, and so people could never type it in correctly. And so all these documents ended up going to the wrong places. So you need to think about that when you're creating the email box. And then we were one of those trials that sort of got stuck in the change from the DEP database to the merge database, and that was a bit challenging for us. As far as the data management goes, we used our clinical folks, and it was really important to help design the ECRF, especially with all the time points we had with the cath lab data we were collecting. We did have a very inexperienced sponsor. They had people from all over the place, but they were really tiny, and they hadn't ever done a big trial like this. So one of the things that they did was they picked out their IVRS and their lab vendors, but they never asked us anything. So we ended up with this IVRS group that created a training database, was in the same database as their live database. So people would go in to try to learn how to randomize a patient, and then they would randomize a pretend patient. So Lisa was constantly backing those people out of the system. We also had a lab vendor that the contract was held with the sponsor, and they didn't put any QC effort into their budget. And so they didn't go in when things weren't there or things didn't make sense and post any queries. So our data team had to kind of come in and do that work for them, which we had not planned to do nor had budgeted for. We also had a brand new angiographic core lab that was at Mount Sinai, and clinically they were very strong. Unfortunately, they weren't expecting the volume that started coming through the doors. They had some technical challenges with some of the systems that we were using, and they had a lot of turnover. So although they did a good job, they had some struggles too. And then at the very end of the trial for the data, we had to send the ECRF data to each of the sites on disks, which would have been fine if we had just ended in a normal manner. But because we ended so early, we didn't have site management people at those sites anymore once we finally got the data on the disk. So we probably need to think about a backup plan for that at the beginning of our trials. From the safety side, as I said, the sites were unblinded, so they knew what the patients had received, and then if they had an SAE, they had to report it in a blinded fashion so that we didn't get unblinded. And so that was really a challenge for the safety team that did a great job. We also had some language in our contract that was very vague, and once we finally worked out what the specs were for us in Parexel, it didn't necessarily match our contract. So I would suggest we sort of put vague language in there if we can. So one of the things is you really need to think about what kind of reports you need for safety, who needs to get them, how frequently you need to get them, and how these SUSARs, SAEs, and allergic reactions are going to be both reported and distributed. So the site management group and monitoring teams created a targeted SDV process, and everybody worked together to create what key fields on the ECRF were going to be looked at and monitored, because we didn't monitor the whole ECRF. As I had sort of alluded to earlier, the con meds, it's challenging when you are saying to a site you need to collect all the meds that go up to the time of PCI for 48 hours before that, from the end of PCI, et cetera. And so it's a challenge to try to go back and document that those things were done. So I know that the monitoring team did struggle a little bit with the way that the ECRF was designed. We had a bunch of site management organizations, as you saw. It's really important to try to consolidate systems so that the people who are monitoring are also the people who are familiar with those systems that are going to track the visits that are being done. And once we were told that we had to close the trial out, it was a challenge to have to shut down these sites really quickly. So we used risk-based monitoring for this particular project, and it really is a collaborative effort to try to get everybody to give input into what's going to be monitored, because we're not monitoring everything. And I think when sponsors hear risk-based monitoring, they think, oh good, we were planning to do 1,000 visits and now we're only going to do 500, so we're going to save all that money. But you need to also remember that there's statistical effort that has to go into that too. If you're not going to be out at sites, you need somebody who's going to program the reports that can provide the look for patterns and things like that, so that those visits don't have to be done on site. So we developed a set of critical variables, and we called them the errors that matter. And then Jen put together a random selection program for us. We identified patients and visits that we thought were going to be important ones to look at, and what kind of patterns we were going to recognize, and then figure out what the action plan was. When you had that pattern show up, what are you going to do, and how are we going to mitigate that process? And it truly was dynamic. You can do it at the beginning, but you've got to be looking at it throughout the life of the trial so that it stays up to date, and you're putting that information you gain back into the system. We also created this targeted SDV tool, which took a ton of time at the beginning, but it worked out really well to identify the variables that were important for monitoring. And just a couple quick thoughts about project leadership. For the project leadership role, we had to do the typical plans that you do, the risk management plan, the communication plan. But for this trial, we had to also create a blinding plan. So we had to talk about who would be blinded, who would be unblinded, how we could guarantee that those people didn't get the information. If somebody did get information, what had to happen next? And we also created a quality by design document. And this was kind of painful at the beginning, but it ended up being a really good exercise because it made us think about what did quality look like for this trial? And what were the risks to not having quality, or what do we need to implement to make sure that we had quality? And then how could we look at those things, how could we monitor them, and what would we do if we met some threshold? So it took a bit of thinking at the beginning, but looking back, it really was very helpful. We also had this tiny little sponsor that said at the beginning about 4,000 times, we don't want any change orders. And so it was almost like running a government grant where they give you your amount of money and you've got to think about things and be creative and figure out how you can make it work. We had a bunch of partners, as you saw. Everybody called the trial something different. Some called it Reg 1, some called it Regatta, some called it Regulate. And so you could never figure out, when you're in the system, how you actually got information. And so I think we spent a lot of time working on developing a trusting relationship with all of our different partners, and I think that was instrumental in getting us to close out the trial as quickly as we did at the end. So the key things we felt like that sort of defined our team were they were collaborative, creative, and committed, and I think that's the reason that we were able to do even what we did in a small point of time. And this is a picture of John. We had late-breaking clinical trials at John's house after the database was locked, and so it was really fun. We got to ask questions and make sure the whole team knew what happened with the trial. So I think Tom is next, and he's going to talk about the results.
So it's my pleasure to talk about the Regulate PCI randomized clinical trial. This is the part where we almost made it to the bedside, and which is really a remarkable thing in and of itself, considering Regatta really had only a handful of employees when we started to run a several $100 million potential clinical trial. So this is the study design, and I just wanted to highlight a few things. We tried to make a very large, simple trial. Our inclusion criteria were basically one, and that's that you needed a PCI. There was only a handful of exclusion criteria, and we really hoped that sites, once they got on board, would find this easy to enroll. And I think that's in great part responsible for the fact that this trial was actually ahead of projected enrollment and was under budget at the time that it had to be curtailed. As people have talked about, this was an open label trial. This was done for a couple reasons, but one of the key considerations we had is that people that were getting Reg 1 or bivalirudin would have different sheath pull strategies, especially if they had a femoral approach. So we didn't feel that it was right that in a patient who had a femoral sheath that would require their sheath to be removed 2 hours later if they were randomized to bivalirudin would also have to undergo the same 2-hour waiting period if they were randomized to Reg 1. That wouldn't represent the way the Reg 1 drug would be used in clinical practice and would subject those patients to 2 extra hours of having a sheath in place that didn't need to be in place. And finally, our primary outcome was at day 3 was a composite of 4 ischemic events. We had discussions with the FDA about what the primary outcome should be, how we balance bleeding versus ischemic events. They came back and said at this time they are only accepting ischemic events as a primary endpoint. And the reason that was important is because we really had to make 2 dosing decisions in this clinical trial. One is how you dose the anticoagulant, which is what you face whenever you're studying a new anticoagulant. But unique to this system is we also had to discuss how we were going to dose the reversal agent. And John presented the RaDaR results, suggesting that there was perhaps a slightly stepwise decreased incidence of major bleeding the more you reverse. We talked about 100% reversal in this drug, in this trial, but in the end we elected to do 80% reversal, partly because we felt like, well, if there's some risk of stent thrombosis or causing acute ischemic events by 100% reversal, we would really be penalized based on our primary outcome. I just want to highlight one aspect of the demographics of the trial or the way the outcomes of the trial, which is that if you look at a trial that was 70% North America enrollment, a predominance of which was in the United States, and almost, I think, over 300 patients were from one femoral site in the United States, we still achieved 50-50 balance between radial and femoral access site. And 32% of the overall trial patients, which is exclusively in the femoral access site, also had vascular closure devices. So what this means is that, in essence, only about 1 out of 6 patients had a sheath in place for several hours after the procedure, which is a marked change in the way patients undergo heart catheterizations compared with when Bruce first presented this idea to Bob and Rob many, many years ago. So this is the primary endpoint of the trial, which was a composite of death, myocardial infarction, stroke, and urgent target lesion revascularization at day 3. You see that there's no difference in the overall composite between the 2 groups. And you also see that the endpoint is overwhelmingly driven by the rate of myocardial infarction. And these are essentially all type 4A peri-PCI MIs that are detected by biomarker elevations in the time period around the trial. Time period around the PCI. Interestingly, though, if you look at the rare events of cardiovascular death, PI identified stent thrombosis or intraprocedural thrombotic events in stent thrombosis, you see that Reg 1 is or pegnevicogen really is a very effective anticoagulant with strong trends toward decreased risks of each of these events in Reg 1 treated patients compared with bivalirudin. If you look at the day 30 endpoint, you see very similar results. Again, the overall composite endpoint is very similar. This is still being driven by the preponderance of MI, peri-PCI MI events that were detected biochemically. And again, you see that stent thrombosis shows a numerical increase in the bivalirudin group, which is now statistically significant. Now, if you look at bleeding, we saw that Reg 1 use was associated with an excess of bleeding events, whether you look at major bleeding as defined as BARC 3 and BARC 5 events, or if you look at all bleeding events as a composite of BARC 2, 3, and 5, with the minor bleeding events reaching statistical significance, and very similar results were seen at day 30. This is a depiction of the enrollment in the trial, which really started in late 2013 and continued up till now, almost a year ago in 2014. Each of the stars is an allergic event that I was notified about on the helpline, which, as you see, as enrollment starts to pick up, occurred more frequently. I think we could have a whole talk about the DSMB approach to this issue during this trial. If you look at the allergic incidence during the trial, assuming that all the events are in the Reg 1 group, which is what we were all obviously afraid of, you see that the incidence of allergies during the course of the trial was remarkably consistent at about 0.6%, which is exactly what we saw in RaDaR. This is now the unblinded allergic results. You see that there's many more allergic events in the Reg 1 arm, but specifically, if you look at serious allergic events, as identified by the site, there were 11 allergies in the Reg 1 arm and only 1 in the bivalirudin group. If you look at severe allergies that were CEC adjudicated, these were adjudicated based on multi-organ system involvement, consistent with anaphylactoid-like reactions. You see that there was 18 in the Reg 1 arm compared with only 4 in the bivalirudin group, and non-severe, more milder reactions were evenly distributed between the two groups. So as has been alluded to earlier, pegnivicogen, and I'll just say a couple things. The allergic reactions, many of them occurred immediately upon studied drug dosing, and many of them were quite severe with hypotension requiring pressor support. Pegnivicogen and all of these occurred, the serious allergic reactions occurred before enivamericin was dosed, so they were all clearly due to pegnivicogen. So pegnivicogen is a composite of an oligonucleotide and a PEG component. The PEG component there is to extend the half-life and keep it in the intravascular space. There's reason to think that neither of these should be very immunogenic. Regatta did an extensive safety evaluation after radar. Oligonucleotides themselves are not immunogenic, and PEG by itself is supposed to be actually immunoprotective. But there was a signal in radar that all 3 of the patients who had serious allergic events also happened to have anti-PEG antibodies prior to study drug dosing, and we just happened to have probably one of the world's experts in anti-PEG antibodies here at Duke in the form of Mike Hirschfeld, who's developed 2 PEGylated drugs that are in clinical use. We did store blood in patients, and we analyzed about all the patients who had any type of allergic reaction within 24 hours of study drug dosing, and patients, about 140 matched controls. And what one sees is that in patients who didn't have an allergic reaction, about 15% of them using this particular assay had anti-PEG antibodies prior to study drug dosing. If you look at the bivalirudin group, you see that of all the patients who had any allergy to bivalirudin, none of them had anti-PEG antibodies. If you look at patients who were treated and had allergies after dosing of pegnivicogen, 10 of the 11 patients who had serious allergic reactions after dosing of pegnivicogen had preformed anti-PEG antibodies, and the only person that didn't was a patient who had an allergic reaction after hospital discharge the following day, which was felt related to ticagrelor. You can also break down these allergic reactions as those that occurred within 1 hour of study drug dosing or more than 1 hour after study drug dosing. And all of the patients who had an allergic event within 1 hour of study drug dosing after receiving pegnivicogen had preformed anti-PEG antibodies. And there's even a signal that patients who had higher levels of anti-PEG antibodies had more serious reactions than those who had lower levels. So I think there's clearly a strong association between preformed anti-PEG antibodies and the allergies that we observed. So the trial-specific conclusions that we drew are that Reg 1 as tested was associated with similar rates of ischemic events with more bleeding compared with bivalirudin. As currently formulated, it's associated with an unacceptably high rate of severe allergic reactions. And we tested, I think, 2 hypotheses in this trial. One is, would active control allow us to use high-level anticoagulation to drive down ischemic events without excess bleeding due to the availability of reversal? And I think while conceptually attractive, the role of this remains undefined. But I think the other thing that we should emphasize more is this is also the first time that factor 9A has been targeted. It was targeted by another company, and the results were that it wasn't an effective anticoagulant, probably due to improper dosing. But our data strongly suggests that targeting factor 9 is an effective anticoagulation strategy, and it appears that there's an association between allergic reactions and anti-PEG antibodies. And I'm going to skip the rest of the few other slides, and we can.
Great. So thanks, Mary Ann and Bruce and Tom. Is on? Anthony, turn this on. Thanks. We're just 3 minutes over, and luckily this is recorded so you can watch it at half speed later. But we wanted to leave a little bit of time for comments, questions, discussion. Yeah, Sunil.
So this is, I mean, it's really impressive how you were able to take this from something that was invented at Duke all the way through the clinical setting. Setting aside the allergic reaction, issues, it seems like you can develop aptamers to almost any target. So without looking retrospectively, the question is why factor 9? I mean, even at the time that this was being developed, the conversation for ACS and PCI was all around factor 10, factor 2. And then sort of related to that is why a 3-day endpoint for a pivotal trial? It just seems odd because the 30-day is sort of the standard and you would accumulate potentially more events at that time.
So, good question about factor 9. So, I breezed through the slides of what we licensed to Regatta. We actually had also made aptamers to thrombin. I showed you a crystal structure of one as well as to factor 10A. We thought about particularly moving the 10A aptamer forward. The challenge with developing the 10A aptamer is it didn't cross-react with animals very well. So the preclinical development was challenging. So it was easier to move forward with factor 9A. The other reason is that there's an argument in the literature that says that 9A is a safer target than 10A. And so that also sort of weighed into the decision. But it was really more a practical reason, I think, which is that was the one we could move more quickly. But we've also made aptamers now to 10A, thrombin, even combination of those, and then we can anticoagulate as well if we fuse those together as well as heparin. So we're interested in moving these very potent, even more potent compounds forward because we can reverse them again.
I mean, one of the neat things Bruce is doing is studying combinations, right? So you could give a 10, a 2, a 9 inhibitor in combination and reverse bits and pieces of them. Regarding your second part of your question, and we had great discussions with the cardiorenal group at the FDA, and what they were looking for is a signal for efficacy, and that efficacy signal is likely, to, as you know, to be really early. We obviously were interested in, does it go away? And so we did collect longer term outcomes. But again, in the interest of designing a really simple trial, almost all of those events were periprocedural. I mean, and many of these patients were in the hospital for a day or not even. So, we really wanted to focus on where we thought we'd see signal efficacy and safety with this approach, and that's early. Chris.
So yeah, thanks. That was a great research conference. Really terrific to kind of see these pieces together. Can you comment? I mean, the implication seems to be that this was a pretty successful foray into kind of a somewhat of a different model than like having a big pharmaceutical company kind of involved before one goes into a phase 3 program. And other than, it's so unfortunate that the allergic thing came up because that obviously kind of distracted or kind of ended it. But other than that, what's the, what would like a business school student who looked this over compared to the usual GSK model or something say about, is this a good way to develop drugs?
So, I don't know the answer to the last part of your question. I mean, Regatta, we had a working relationship with the Regatta team from its inception. I mean, so from Regatta's inception. I mean, and through 4 or 5 trials, and actually the entire personnel in Regatta turned over. We had 2 CEOs, we had 3 chief medical officers, we had, so it was multiple people over this time. Up through, I mean, Radar was a stretch for them financially. And that was all venture funded. They then tried to partner with Big Pharma unsuccessfully to do the phase 3 trial. And then they had a CEO and a chief medical officer who had Big Pharma experience, and they thought they could go out and raise the money. So a bunch of this trial was funded by the Russian government, for better or worse. Regatta went public right before Regulate, and so a lot of it was people's stock investments in Regatta, and they came up, and then we also were on a tighter budget. I think they didn't have a lot of extra money. They had the money to run the trial, but not a lot of extra money. They had a small team with less experience. That has pros and cons. And then as soon as the trial was stopped, Regatta evaporated. I mean, within 3 to 6 months, today there is no Regatta. It's gone, and all the people are gone. I think their third CEO is still there, but at a new company focused on NASH and hepatitis. So, and I've connected them with Andrew Muir. But I think, so it's a really different experience than working with a big pharma Some of the big pharma companies they talked to said, you guys are taking all kinds of risks. You should be doubling the size of your trial. You should be collecting all these unnecessary safety events. You should be doing all the things that we think are largely a waste of time, but that big pharma does because they have more money to throw at a project. Yeah.
For John and Tom, I'm interested to know whether you guys believe that this is an asset still and potentially recoverable. So is there enough in the clinical data, for example, that you might say there's a way out of this, reformulate the drug, use testing, for example, to try to identify the people that might be at risk? Is there enough here to justify that? That's an important question for Duke, it's an important question for the founders, et cetera. So that's one question that I have, but start with that one.
Well, I would say clearly not for this indication. I mean, we had discussions along the line about whether or not anti-PEG antibodies would be a co-diagnostic test that could be used to predict who's going to develop an allergic reaction. I mean, we have one of the world's experts in Mike. We're convening the allergists that were involved with this to write this up. I think the data that we have clearly suggests that if you have anti-PEG antibodies to a significant level, you are at risk of having these reactions, and one could argue maybe that if you don't have those antibodies, you could get this drug safely. That would have to be done in a different setting, not an acute PCI setting where we're too impatient to wait around for an antibody test to come back to do a PCI on somebody. But maybe in another setting where the need for anticoagulation is more elective, like someone undergoing TAVR, someone undergoing heart surgery, and reversal is particularly attractive, maybe there could still be some use of a co-diagnostic, and then Bruce can talk, but there are other ways to formulate these drugs. PEG has been used for a while because of its...
Do you believe that you need a 24-hour half-life for that indication?
Yeah.
Or could you do with significantly less, like cholesterol, for example, that has a 2 to 4 hour half-life?
Yeah, I guess I don't think you need a 24-hour half-life.
Right.
Yeah, I mean, we had some of these discussions with Regatta, and right, so I think the drug works to anticoagulate people and reverse. Whether it's better than existing parenteral anticoagulants is I think an open question, and whether there's space in a crowded marketplace for another reversible anticoagulant is an open question. What Tom mostly talked about is that, yeah, the drug could be reformulated with other means to give it a longer half-life. It might not be quite as long as PEG. But the question is, it's still a couple $100 million development path to get from there to a commercial compound. And in light of the bleeding and efficacy outcome data we have, is that something somebody wants to take on? And that's really where I think Regatta felt there was a big challenge to figuring out how to move this forward. Yeah, I mean, it's interesting. So remember, Duke has, I mean, and Marianne alluded to this on her slide, so Duke has equity in some small amount of equity in Regatta. This compound was discovered at Duke. So when we were going and talking to the FDA, Duke is viewed as a sponsor of this. Now that equity is firewalled, so I don't know how much it is or what it's worth, and nobody on the team did. But we had a lot of interesting discussions about how to run a trial with a company that Duke owns a small portion of.
Let me press a little harder on that. So a reversible anticoagulant seems to be like a very important advance.
I think it's still intuitively really interesting and really promising. I mean, what we didn't show, and there are lots of limitations to the trial, is that it has any measurable benefit over existing anticoagulants. And so now we gave a certain dose of the anticoagulant, we reversed it 80%, not 100%. We stopped the trial early because of allergic events, and so we're underpowered. We might have been measuring the wrong ischemic events. All those periprocedural MIs might not be affected as much by an anticoagulant. So there are lots of things where one could redesign a trial that might demonstrate a bigger benefit. But as I said, it's a long and expensive road to go down into a crowded space. These reversible, I mean, Regatta had a cardiac surgery program that was early as well, because cardiac surgery is an obvious place where a reversible anticoagulant might be attractive. That might be a place where a cocktail of anticoagulants all being reversible might be even more attractive.
Extension of sort of the question, is there, are there particularly subsets, so we don't have to do $100, $200 million trial? So, for example, in cardiopulmonary bypass, we think about a hit patient. Is there a subset of patients where you can say you can go for, bivalent or heparin are not, appropriate, so that you can think about a development path where you don't have to do the type of trials we were looking at?
Yeah, I think potentially, although it's not one trial that has to be done again.
Yeah,
It's
you have to go back
several.
if you're gonna
Yeah,
reformulate.
it's several trials. You have to do again.
I'll just make one comment to both Chris's question and your question, is that it's amazing to me, like, I was kidding Bruce about this taking 15 years, and it took 15 years and things went almost all perfectly all the way through. But it's amazing how much the landscape has changed. And one of the things that the small company model did do is there was a large delay between radar and regulate, primarily due to money concerns. And I think that's probably gets involved in other programs as well, but that might have speeded up the process if a large pharma company had been involved earlier, because just the whole radial landscape and everything has changed so much since the time Bruce had his initial discussions.
And one last question: if I want to do a trial now, with which hopefully we want to do every trial this way, with a monitoring approach that's kind of rationally based on focusing on key quality elements and risk. How are we doing at DCRI on integrating lessons from an experience like this into an institutional approach, so that when I go to our monitoring group, that I can be assured that kind of the wisdom from this trial is effectively transferred to the next project?
Yeah, you should ask them, did they come to research conference? No, I think, we have a number of initiatives related to risk-based monitoring that I don't think we have a perfect structure. I think we have a number of initiatives related to risk-based monitoring, and there's more sharing, I think, of ideas between ideas and lessons learned between trial teams and various groups. Risk-based monitoring is a huge, broad, sort of overarching concept, and we had a lot, as Marianne said, we had a lot of discussion up front about, okay, what's important in the trial, how do we make sure we're focusing on that, and that was all facilitated by having a small sponsor without a lot of money. I mean, they were interested in ways that we could do this trial simpler, and they had no one at the sponsor who was driving the monitoring discussion either. So we were really the drivers. We were in the driver's seat there, and because of that, had more ability to implement what we thought was a rational approach. And I think we did. I think we wrote the protocol. I think we designed a simpler trial and ran it in a fashion that was probably half the cost of what some of your big pharma friends would have run a trial like this for. So thank you, everybody. Thank you, Bruce and Marianne and Tom. It's been and for everybody else who was involved in Regulate.